The overall goal of the following experiment is to estimate viral titers of a luciferase-tagged virus using a high-throughput quantification method. This is achieved by transferring the supernatant of the samples to be titered, as well as a viral standard curve, onto a permissive cell line to allow for luciferase expression. As a second step, resizurin can be added to the original samples, which will be used to assess cell viability. Next, following an appropriate incubation time, luciferin is added to the cells in order to quantify bioluminescence. The results show the amount of virus in the sample based on luciferase expression.
The main advantage of this technique over existing methods, like plaque titering, is that a large number of samples can be processed quickly and simultaneously.
This method can also be extended to non-plaquing luciferase-expressing viruses, since it is not dependent on plaque formation.
A readout of cell cytotoxicity can easily be incorporated into the workflow and may be useful in the context of a drug screen.
Demonstrating the procedure will be Vanessa, Ramya, and Colin, 3 graduate students from my laboratory.
First, perform infections using VSV delta 51 expressing firefly luciferase in 96-well tissue culture plates. Supernatants from these plates will be titered after an appropriate incubation time. 24 hours prior to titering, prepare a suspension of viro cells at a concentration of 2.5 times 10 to the 5th cells per milliliter in DMEM containing 10% FBS, 30 millimolar heaps, and 1% penicillin streptomycin. Seed 2.5 times 10 to the 4th cells in 96-well white solid flat bottom plates using a microplate dispenser. To prepare the cell suspension,